Transcranial direct current stimulation and major depressive disorder by Alan Blydenstein. In this presentation, I'm going to outline and overview what major depressive disorder is and how it affects the brain and how direct current stimulation is an effective treatment tool to treat patients with MDD. Also going to talk about how astrocytes play an important role in TDCS for treating MDD, and I'm going to also go over an experiment conducted by Monet and colleagues, a controlled experiment with mice, how they induced depression in these mice and then used TDCS to see what the results were. And also how TDCS promotes plasticity and increased excitation with neurons. Transcranial direct current stimulation is a form of non-invasive neurostimulation that delivers a constant low-level current to the brain through the use of electrodes typically placed on the scalp. The neuromodulation technique uses a steady current between 1 to 2 milliamps delivered through electrodes to targeted regions of the brain to modulate brain function. Using different electrode configurations, TDCS can be used to either excite or inhibit neuronal activity in the targeted region. The current used in TDCS is not strong enough to trigger an action potential, but rather affects the neuron's resting membrane potential. When applying a nodal stimulation, the electrode is used to facilitate depolarization of the neurons, resulting in excitatory stimulation. However, during cathodal stimulation, the electrode is used to induce hyperpolarization of the neurons, resulting in inhibitory stimulation. An increasing number of human studies have reported that TDCS enhances the brain functions, including motor learning and memory. In addition to memory and learning enhancements, TDCS has been probed for its therapeutic potential in various psychiatric and neurological conditions. Of these, the most intensively examined use of TDCS in psychiatric conditions is major depressive disorder. Major depressive disorder is a depression categorized of being long-lasting over the course of months and years and incapacitates one's physical and social abilities. The alleviation of depression symptoms helps improve the quality of life of patients who suffer from MDD, while psychotherapy and drug treatment such as fluoxetine have been shown effective for some patients with severe depression. Statistics show that this is only effective in about 50% of people with MDD. Transcranial direct current stimulation has proven to have much higher results as an effective treatment. MDD is one of the most pressing public health problems in the world, where patients present with their depressed mood, loss of interest or pleasure, decreased energy, feelings of guilt or low self-worth, disturbed sleep or appetite, and poor concentration and cognitive impairment. The main physiological feature of MDD are an imbalance of left and right prefrontal cortex and dysfunctional cortical subcortical neural networks. Hypoactivity of the left dorsolateral prefrontal cortex and hyperactivity of the subcortical and limbic regions are key features in MDD. What this means is the left side of the brain is not communicating with the right side of the brain properly, and this is where TDCS comes in. It directs current flow from the right side of the brain through the left side of the brain, and it promotes excitation and neurostimulation so the neurons start to fire more effectively. And this promotes the plasticity and neural activity in the brain. Astrocytes are glial cells that interface synapses and blood vessels, making them an attractive target of therapeutic interventions by systemic drugs. Astrocytes maintain the extracellular environment by removing neurotransmitters and absorbing excess potassium. Astrocytes are also employed in mediating energy substrate transfer from the bloodstream to neurons. In addition to these so-called classical functions of astrocytes, gliotransmission secretion of bioactive molecules from astrocytes has been proposed to be a key mechanism to modulate synaptic transmission and plasticity. This slide depicts how TDCS alleviates a mouse model of depression by chronic restraint stress. Monet and colleagues did an experiment on mice where the mice were exposed to chronic stress to induce depression, and the immobility times were recorded. The graph shows how by using TDCS, this greatly reduces the immobility times of the mice. From the observation that an anodal direct current applied on the peel surface of the rat brain increases cortical neuronal discharge, Bindman and colleagues found that TDCS has been recognised to elevate the excitability of neurons in the anodal area. A great amount of literature seems to support the notion of anodal TDCS, which causes a subthreshold depolarization of neuronal membrane potential, resulting in an increased probability of neuronal discharge and/or synaptic transmission. Following the enhancement of motor-evoked potentials by TDCS in human subjects, rodent slice physiology studies have shown that synaptic plasticity was enhanced in slices that were prepared from animals that received TDCS. In conclusion, although the effectiveness of TDCS as a treatment for depressive disorder is still controversial, analysis of current literature indicates that TDCS is effective in the treatment of MDD. Monet and colleagues' mouse study indicates that TDCS activates cortical astrocytes via noradrenergic receptors in the sensory cortex. Moreover, the activation of astrocytes observed by calcium ion elevations is highly influential to the synaptic plasticity of depression-like behaviour. Transdirect current stimulation targeted to the prefrontal cortex, a common experimental procedure for treating depression in patients, might also activate astrocytes by dopamine, since the prefrontal cortex consists of many dopaminergic fibers. Monet and colleagues also speculate that patients with drug-resistant depression may exhibit compromised calcium elevations in cortical astrocytes in response to TDCS treatment. It is certainly a challenge to demonstrate the same experiments on mice as we do in humans, as there are many ethical concerns, hence why us humans tend to experiment on the animals first. The use of TDCS is still controversial, and the research in this field is still developing. There is a lot more we need to learn about TDCS and how effective it is before it becomes much more widely used method to treat MDD.